Hello everyone, myself Manju Sharma from Suresh Kanvyar University, Jagatpura, Jaipur, and the department is School of Agriculture. Today we are going to discuss about disease of cotton. Uh, Disease of cotton, in which the first disease is the Anthracnose disease, and the causal organism is Coletotrichum capsicum. The pathogen infects the seedlings and produces small, reddish, circular spot on the cotyledons and primary leaves. So the lesions develop on the collar region. Stem may be graded, causing seedling to wilt and die. In mature plants, the fungus attacks the stem, leading to stem splitting and shredding of bark. So basically, this disease will be in mature plants, and so due to the attack on the stem by the fungus, the stem will be spreading, and then it will be shredding of bark. The most common symptom is boll spotting, a small water shock, circular, reddish brown, depressed spots appear on the balls, and the lint is strained to yellow or brown, becomes a solid brittle mass of fiber. So the infected balls keys to grow and burst and dry up prematurely. So basically, the main symptom which will be shown on the balls of cotton. So different types of sports will be there. So these are the main sports and the main symptom, and we can say that there is the ball spotting. And basically, due to these sports, the infected ball which will be burst prematurely or they will be dry. So automatically the yield of the cotton, it will be less. Pathogen, the pathogen forms large number of acervuli on the infected plant. The conidiophores are slightly curved, short and club shaped. The conidia are high aligned. High aligned, that means like different types of spots are there. So basically, the color of the spot which will be dark at the periphery and in the mid also, but they will be high aligned, they are highlight and facilitate bond single on the conidiophores. So the numerous black colored and thick walled seed are also produced in acerulus. Basically, in different types of fungus, the name of the spores will be also different. Okay, like in some acervuli will be there, in some conidiophores are there, in some fungi basically Puccinia are there. So these will be different. Their favorable conditions like prolonged rainfall at the time of ball formation and close the planting. So these are the favorable conditions for the pathogen. Disease cycle, the pathogen survive as dormant mycelium in the seed or as conidia or the surface of seed. The pathogen survive as dormant mycelium in the seed or as conidia on the surface of seeds for about a year. The pathogen also preparates on the rotten balls and other plant debris in the soil. because like any pathogen, those can easily survive on the plant debris if they are available in the field, because they will be gets their nutrient from the particular uh, like they can get their nutrients from the crop residues, so they will be live alone. That's why during the management we always include that we have to apply field sanitation, because if crop debris will be not there, in that case, due to the off season of the main crop, the particular pathogen, those will be also lost after some time, and they will be not live there, or they will be not alive, because there will be the lack of nutrients. But if crop nutrients will be available, crop debris are there, then they can get their nutrients from them easily. The pathogen also precipitates on the rotten balls and other plant debris in the soil. The secondary spread is by airborne conidia, and the pathogen also survive in the weed host via Aristolochia, Brachytata, and Hibiscus diversifolius. Like these are the symptoms. Here you can see on the symptoms on the leaf and also on the ball. Okay, so this ball, after showing the symptom, basically it will be the balls will be ruptured prematurely and the production will be less. Their management, like treat the delented seeds with carbendazim or carboxyn or theorem or captain at 2 gram per kg. Then remove and burn the infected plant debris and bolls in the soil. Rub, rub out the weed host. Spray the crop at ball formation. Stays with mancozeb like 2 kg or copper oxychloride 2.5 kg or carbendazim 500 gram per hectare. So basically these are the chemical treatment by which we can manage the disease. After that, bacterial blight disease. Bacterial blight, that means the causal organism is bacteria, not the fungus. After that, diseases of green gram. Green gram, in which the first disease is the powdery mildew disease, and the pathogen is Erisiphe polygoni. So basically, the powdery mildew disease, the main symptom or the common symptom of this disease, like powder-like appearance will be there at the lower surface of the plant, and after some time they will be make a colony, and the colony will be white in color, or it's look like a powder like appearance. Okay, that's why the name given powdery mildew disease. Downy mildew disease is also there, but in downy mildew basically the spots on the leaves will be like lightly, and they will be show yellow in color. Or powdery mildew, basically white powder like structure will be there, and for the downy mildew disease basically yellow color spots will be there. Symptoms: powdery mildew is one of the widespread disease of several legumes. In green gram, white powder patches appear on leaves and other green parts which later become dull and colored. So these patches gradually increase in size and become circular covering the lower surface also. So when the infection is severe, both the surface of the leaves are completely covered by whitish powdery growth. So we can easily identify the powdery mildew disease, or basically it is a major disease. It's like we can say that in all types of plant, or in mostly plant, the powdery mildew disease is there, and due to their whitish appearance, the name given powdery mildew, because powdery like appearance are there. Severely affected, parts get shriveled and distorted. In severe infections, foliage becomes yellow, causing premature defoliation. So the disease also creates forced maturity of the infected plant, which results in heavy yield loss. Like the pathogen is ectophytic. Ectophytic, that means they will be attached to the surface of the plant, and then it will be cause the disease. Okay, ectophytic, that means they will be absorb the nutrients from the surface of the plant. These are not endophytic. Endophytic, basically the pathogen which will be like invade inside the plant tissues and then get their nutrients, and we can say they will be make the contact with internal organs. Okay, but the pathogen is the ectophytic, spreading on the surface of the leaf, sending hysteria into the epidermal cells. So the conidiophore arise vertically from the leaf surface, bearing conidia in short chain. So conidia are high align, thin walled, elliptical or barrel shaped or cylindrical, and single celled. So later in the season, Cleistothecia appear as minute black globose structure with myceloid appendage. Each cleistothecium contains 4 to 8 asci, and each ascus contains 3 to 8 ascospore, which are elliptical, high aligned, and single celled. Favorable conditions for the pathogen. The pathogen has a wide host range and survive in ideal form on various hosts in off season. Basically, the pathogen, those have the wide host range, then they can live in any kind of conditions. Okay, because if there is no availability of a particular host, then they can easily survive on the other host. Secondary spread is through the airborne oidea produced in the season. Disease cycle. The fungus is an obligate parasite and survive as Clostridium in the infected plant debris. Primary infection is usually from ascospore from prenating Clostridium. The secondary spread is carried out by the airborne conidia. The pathogen is the fungus is the obligate parasite. That means they always require a living host. And the fungi which have a wide host range, that's why they can absorb their nutrients and they will be feed at any host. Their management, like use resistant varieties. The seed must be sown early in the month of June to avoid early incidence of the disease on the crop. So basically, if like in the June, later in the June or early in the July, if seed showing will be there, in that case the effect of the pathogen will be more, or maybe it will be aggressive or virulent. But if in the month of June or in early June, the seed showing will be there, in that case we can overcome the effect of the pathogen. Spray of carbendazim 500 gram or vegetable sulfur 1.5 kg or tridmor 500 ml per hectare at the initiation of disease and repeat 15 days later. Then the next disease is anthracnose disease. The pathogen is Coletotrichum lindemuthenium, and during the sexual stage it will be called Glomerella lindemuthenum. The pathogen is the same, but their sexual stage or their vegetative stage both will be different. Symptoms are the disease appear on all aerial part and at any stage of plant growth. Like this disease which can cause the effect or the symptom on the any aerial part. Aerial part, that means like except the root, the pathogen which can cause the disease or the symptoms may be appear on any part of the plant. So these are circular, black, succulent spot with dark center and bright red orange margins on leaves and pods. In severe infection, the affected parts whiter off. Seedling get blighted due to infection soon after seed germination. Pathogen. The disease appears on fungus mycelium is septate. Mycelium, that means branching of the hyphae, and septate, that means the hypha which will be divided into different segments. High aligned and branched. Conidia are produced in acervuli, arise from the stroma beneath the epidermis and later rupture to become erupent. A few dark colored septate setae are seen in the acervuli. The conidiophores are high aligned and short and bear oblong or cylindrical high aligned, thin walled, single cell conidia with oil globules. The perfect stage of the fungus produces perithecia with limited number of asci, which contain typically 8 ascospores, which are one or two cell with a central oil globule. Their favorable conditions, like the disease is more severe in cool and wet season. Basically, the pathogen which can survive, or it will be survive at lower temperature. Disease cycle. The fungus is seed-borne and cause primary infection. It also lives in the infected plant tissues in soil. The secondary is spread by airborne conidia produced on infected plant parts. Rain splash also help in dissemination. Their management, like hot water treatment at 54 degrees centigrade for 10 minutes, because like the low temperature which will be favorable for the pathogen. So if we are doing the management, then if we will give the treatment like hot water treatment, and the like it should be boiled, and it will be arise at 54 degree. Okay, so if we will give the hot water treatment at 54 degrees centigrade for 10 minutes, use disease free seeds, hollow crop rotation, remove and destroy infected plant debris in soil, then treat the seed with carbendazim at 2 gram per kg, and spray of carbendazim 500 gram or mancozeb 2 kg per hectare soon after the appearance of disease, and repeat after 15 days. Then the next disease is the leaf spot disease, and the pathogen is Sarcospora cancescens. Symptoms are like this is an important disease of green gram, is usually occurs in severe form, causing heavy losses in yield. Spores produced are small, numerous in numbers, with pale brown center and reddish brown margin. So, the under favorable environmental conditions, severe leaf spotting and defoliation occurs at the time of flowering and pod formation. Pathogen. The pathogen produce clusters of dark brown septate conidiophores. The conidia are linear The conidia are linear, high aligned, thin walled, and 5 to 6 septate, favorable conditions, and high humidity favors disease development, because if more humidity will be there, then the pathogen which will be spread or it will be survive there for long time. Disease cycle, like the fungus survive on diseased plant debris and on seeds. The secondary spread is by airborne conidia. Their management: cultivate resistant varieties, intercrop the mung with tall growing cereals and millets. We have to follow clean cultivation, use of disease-free seed, maintain low crop population density, and wide row planting. So the crude extract of cassava, garlic, and ginger are applied for controlling the disease effectively. Mulching reduces the disease incidence, resulting in increased yield, and spray of mancozeb 2 kg per hectare or carbendazim like 500 gram per hectare. Then the next is the rust disease, which caused by the Euromyces fascioli, and Euromyces appendiculatus. Symptoms are like the disease appears as circular reddish brown postulates which appear more commonly on the underside of the leaves, less abundant on pods, and sparingly on stem. So, when the leaves are severely infected, both the surfaces are fully covered by rust postulates, shriveling, followed by defoliation, resulting in yield loss. The pathogen is it is autoceous, long cycle rust, and all the spore stages occur on the same host. Like pathogen is autoceous. Autoceous, that means we can say that a parasite which completing the entire life cycle on a single species of host. Okay, like we can say that as compared to heterogeneous of plant which having male and female reproductive organs on the same plant. Or basically, autoceous, that means whenever the any host, sorry, any parasite Those complete their life cycle within a single host. Okay. The uridospore are unicellular, globose or ellipsoid, yellowish brown with cannulations. The teliospores are globose or elliptical, unicellular, pedicellate, cashew nut brown in color with warty papillae at the top. Then yellow color puccinia appear on the upper surface of leaves. Orange color cupulate Asia develop later on the lower surface of leaves, and the spores are unicellular and elliptical. Favorable condition for the pathogen like cloudy humid weather, temperature 21 to 26 degrees centigrade, and nights with heavy dews. So basically for the fungus, the optimum temperature is between 20 to 30, in which like all types of fungus may be grown. Disease cycle, the pathogen survive in the soil as teliospores and as uridospore in crop debris. Primary infection is by the sporidia develop from teliospores. Secondary spread is by wind-borne uridospore, and the fungus also survive on other legume hosts. Their management, remove the infected plant debris and destroy. Spray of mancozeb 12 kg or carbendazim 500 gram, sorry 1 to 2 kg or carbendazim 500 gram, or propiconazole is 1 liter per hectare or 1 kg per hectare immediately on the set of disease, and repeat after 15 days, and we have to use the tolerant varieties. The next disease is the dry rot disease. Dry root rot, which caused by Rhizoctonia bataticola. Their puccinudial stage is Microformina flaciolina. The symptoms are like the disease symptom starts initially with yellowing and dropping on the leaves. The leaves later fall off and the plant dies within week. The dark brown lesions are seen on the stem at ground level, and so the disease symptoms start initially with yellowing and drooping of the leaves. The leaves later fall off and the plant dies within week. So dark brown lesions are seen on the stem at the ground level and bark shows shredding symptoms. The affected plants can be easily pulled out living dry, rotten root portions in the ground. So the affected plants can be easily pulled out, leaving dry, rotten root portion in the ground because the disease is the root rot disease. So basically, the main infection in the root, and whenever we will be plugged out, then it will be easily. So the rotten tissue of stem and root contain a large brown root contain a large number of black minute sclerotia. So the fungus produce dark brown septate mycelium with constrictions at hyphal branches, minute dark round sclerotia in abundance. So the fungus also produce dark brown globose osteolated. The fungus also produce dark brown, globose, osteolated Puxinidia on the host tissue. So the Puxinidio spores are thin walled, high aligned, and single cell and elliptical. Favorable conditions like day temperature of 30 degrees centigrade, which prolonged dry season followed by irrigation. Disease cycle. The fungus survive in the infected debris and also as facultative parasite in soil. So the primary spread is through the seed-borne and soil-borne sclerotia. The secondary spread is through airborne puxinidiospores. Management like treat the seeds with carbendazim theorem at 2 gram per kg or pellet the seeds with trichoderma viride at 4 gram per kg or pseudomonas fluorescens 10 gram per kg of seeds and apply the farmyard manure or green leaf manure, that is Glaricidea maculate at 10 ton per hectare or neem cake at 150 per hectare, 150 kg per hectare. So this is all about the diseases of green gram. Thank you. ये बना लो फिर बता रहा हूँ. Cultural method of plant disease management. So, firstly, general principles in which like avoidance, exclusion, eradication, protection, disease resistant, that is also called immunization. So, let's come avoidance. Avoiding disease by planting at times when or in areas where inoculum is ineffective due to environmental conditions or is rare or absent. Exclusion of inoculum, preventing the inoculum from entering or establishing in the field or area where it does not exist. Eradication, like reducing, inactivating, eliminating, or destroying inoculum at the source, either from a region or from an individual plant in which it is already establishment. Then protection, preventing infection by creating a chemical toxic barriers between the plant surface and the pathogen. Disease resistant, that is also called immunization. Basically, it preventing infection or reducing effect of infection by managing the host through improvement of resistance in it by genetic manipulation or by chemical therapy. Then avoidance of the pathogen. These methods aim at avoiding the control and the contact between the pathogen and susceptible stage of the crop. So this is achieved by proper selection of geographical area, wet areas and dry areas. Secondly, proper selection of the field in which the sick field or same crop year after year. Then, adjusting the time of sowing, avoidance of favorable environmental conditions for pathogen, disease escaping varieties in which resistant or specific growth characteristics, and proper selection of seed and planting material in which seed and seedling material from healthy sources. Then exclusion of the pathogen. So these measures aim at preventing the inoculum from entering the establishing in the field or area where it does not exist. Different methods of exclusion are like the seed treatment, seed infection, seed inspection, and certification, and the crops grown for seed purpose are inspected periodically for the presence of diseases that are disseminated by seed. Necessary precautions are to be taken to remove the diseased plant in early stages, and then the crop is certified and the disease free. This practice will help in the prevention of inter and intra-regional spread of seed-borne diseases. Plant quarantine regulation, a legal restriction on the movement of agricultural commodities for the purpose of exclusion, prevention, or delaying the spread of the plant pests and diseases in uninfected areas. Then, eradication. Eradication. These methods aim at breaking the infection chain by removing the foci of infection and starvation of the pathogen. For example, like annihilation of the pathogen from the area by destruction of sources of primary and secondary inoculum, and it is achieved by like rogging. Rogging, that means removal of diseased plants or their affected organs from field, then eradication of alternate and collateral host, crop rotation, crop sanitation, collection and destruction of plant debris from soil, and manures and fertilizers. So, the deficiency or excess of a nutrient may predispose a plant to some diseases. Mixed cropping, root rot of cotton, that is Phytotrichum omnivorum, is reduced when cotton is grown along with sorghum. Summer plugging, summer months expose soil to hot weather, soil amendments like application of organic amendments like sawdust, straw oil cake, etc. effectively manage the disease. So they also allow growth straw, oil cake, etc. effectively manage diseases. They also allow growth of beneficial microorganisms, thus the suppression of pathogenic microbes. Then the changing time of sowing, seed rate and plant density, like close spacing raises atmospheric humidity and favors the sporulation by many pathogenic fungi. Seed rate and plant density, close spacing raises atmospheric humidity and favors the sporulation by many pathogenic fungi. Irrigation and drainage, the amount, frequency, and the method of irrigation. Protection, use of chemicals for the control of plant diseases to stop the pathogen from entering the host or checking the further development in already infected plants. The chemicals used are called proctants, therapeutants, and therapy. Therapy means cure of disease in which the fungicide is applied after the pathogen is in contact with the host. Chemicals used are called therapeutants, fungicide, any agent or chemical that kills the fungus. Fungicide, that means some chemicals which do not kill fungi but simply inhibit the fungus growth temporarily. Antisporulent, the chemical which inhibit the spore production without affecting vegetative growth of the fungus. So basically, these all types of chemical which used in the production. So the therapeutants, the chemicals which use in the therapy, the fungicide which kill the fungus, the fungicide which stop or inhibit the growth of the fungus, and antisporulent which inhibit the spore production. Thank you.